Hello everyone. Today we will be discussing about Vibrio cholera. Okay, so first I want you to know that Vibrio cholera is a gram negative. It is a salt loving. It's an alkali loving comma shaped bacteria. It's a gram negative bacteria. It loves salt and it can thrive in alkali environments. Before I proceed, I want to tell you a small story. So this is Mr. John Snow. So he was a medical student in London in the year around 1847, 1857. So he observed that in his locality there was an outbreak of cholera. So what he did was he plotted out the cases on the map using a dot technique. As you can see, these indicate more cholera cases. And all this pointed out that this pump, that's a Broad Street pump, was a source of all the infections here. Classification. So first there are three types of classification. First is serogrouping, serotyping and biotyping. So serogrouping. So first before that I want you to see this is a gram negative cell wall. As you can see, this is a lipid membrane, this is a lipoteichoic acid, this is a cell wall, and from the cell wall we can see a polysaccharide, and from which we can see an O specific side chain. O specific side chain is also called O antigen. So this O antigen has many variations. Since it is a gram negative bacteria, it has many variations. See, it is part of the cell wall. So it's called a somatic antigen. So using that antigen based on its variation, we can classify in serogrouping. Serotyping. What is the difference between serogrouping and serotyping? Here we use the type of antigen. Suppose we can use LPS, we can use O antigen, so on. But here we use O antigen. If it is present, it's called based on O antigen, we classify it into O1, O139. Serotyping, it is a minor variation in the O antigen. So the O antigen will be present, but there will be slight variation. Suppose if it is N and N, it might be M and N. So there might be a slight variation. Next is biotyping. So based on the biochemical or the physiological difference, it is differentiated into types. Now we will see about the serogroups. So based on the O antigen present in the cell wall, where is the O antigen? It is present in the cell wall, as you can see. So based on that, we can classify it. So against that antigen, we obviously will have antibodies, right? So these antibodies, also called antisera, are used. So if there is O1 antisera used and it agglutinates, it is O1. If you use O139 antisera, it is O139. So if there is a Vibrio cholera and it is not agglutinated by either O139 or O1, it is called non-O1 or O139. Okay, next we'll go to biotypes. So biotypes, as I already have said, it is based on the biochemical and the microbiological properties. Under the O1 serogroup, we have classical and the Altor type. This classical was responsible for the first 6 epidemics, and this was responsible for the 7th epidemic. It started in the Gangetic Delta, and this started in the Sinai Peninsula. It is slightly less pathogenic, and this is slightly more pathogenic. And this is a newer version, and this is an older version. Now serotyping. Now the O1 serogroup is there, right? So based on the minor variations in the O antigen, we classify it into Ogawa, Hikojima and Inaba. These are African names. I know it is tough to pronounce, but you can just slightly remember. So what is the use of these Ogawa, Hikojima and Inaba? So Ogawa and Inaba, they are like the extremes. So this is at the start and this is at the end of an epidemic, and this is the transition stage. So when an epidemic progresses, it first Ogawa, then it goes to Hikojima, and then it goes to Inaba. There is a transition, and this transition is brought about by RFTB gene. So this is a genetic process. So as the epidemic progresses, due to immune selection, selective pressures, and this RFTB gene, it goes from Ogawa to Hikojima to Inaba. Next, we will see the pathogenesis. So, first, when you eat 10 to the power 8 bacilli enter your stomach, and when does it enter? It's when your acidity is lowered. When is it lowered? When you use antacids or when there is hypochloremia. So what happens is, because when there is extra acid, obviously a bacteria can't thrive there, except Helicobacter pylori, which causes peptic ulcer disease. But here we are discussing about cholera. So cholera comes, and as you can see, it travels from the stomach. After stomach, what does come? Intestines, right? So when it comes to the intestines, as you can see, this is the epithelium, and you can see the mucous membrane, mucous surface. So what this cholera does is, it secretes proteases, proteolytic enzymes, which degrade this mucous layer. The mucous layer is degraded, it uses its fimbriae to attach to the epithelium, secretes a toxin, and this toxin has A and B. So the B toxin enters here, binds to the M1 ganglioside receptors. This is present in the epithelial cells. As you can see, these are the columnar epithelium. So what is the epithelium of the intestine? It is columnar, simple columnar epithelium. In those cells, they have this M1 ganglioside receptor. The B binds to the M1 ganglioside receptor, and it causes the internalization of A protein, A toxide. So A toxide comes and activates the G protein. So what does this G protein do? It has an effect on adenyl cyclase. It uses ATP to, so as soon as it uses ATP, it activates CAMP, and the CAMP activates protein kinase, and this activates membrane chlorine channels. So when chlorine channels are excited, they secrete chlorine into the lumen. Now our body doesn't like this hyperchloridia in the lumen, so it secretes sodium into the lumen to balance out the osmolarity. Now since there is sodium and chloride more in the lumen, water eventually processes out due to osmosis. Now when there is a lot of water increasing, what do you think will happen to the stool? Yes, it will be watery. Okay, and it is a phage control protein. What is the importance of this phage control protein? Because it is used for making non-toxidal bacteria for making of vaccines. Next we'll go to the clinical manifestations. 75% of them are asymptomatic, and another 20% have mild diarrhea, and 5% of them have cholera gravis. It is a very serious situation where there is almost 10% body weight loss in form of water, and the incubation period is 1 to 2 days. So if you consume your food which was contaminated by the cholera yesterday, it will affect you today. So when the patient comes to you, how will the stools be? It will be painless. He doesn't have pain passing it. If he has pain, what can you suspect? It can be constipation, it can be piles, it can be anal fissures. It is voluminous because there is lot of water in the stools. It will be voluminous. And watery, voluminous. Why? Because there is lot of water in the stools. Rice watery stools. It has fishy, inoffensive, no mucus and blood because there is no underlying pathology. It is just a toxoid which affects and causes this diarrhea. There is no mucus or blood. Now, because of this, your body is losing its sodium and chloride. So, now what will happen? And your water also. So, now since you have lost all this, your GFR will be falling. And if your GFR is falling, what will you expect? First, it will activate your RAAS system and it causes thirst. Next, if you lose 5 to 10%, it will cause weakness. Weakness why? Because you don't have adequate blood volume. And tachycardia. Why? Because your body wants to compensate for the low amount of body volume. And hypotension, because there is no body blood volume, so there will be fall in blood pressure. If it is more than 10%, first you go to oligouria and then wrinkled skin and you can even go to coma. About the epidemiology. Its homeland was the Indo-Gangetic Delta, that is Bangladesh, and the Indian state of West Bengal. So there were 6 pandemics, 1 to 6, caused by classical type. It was between 1817 and 1923. Epidemic 7 was caused by the L-Tor variant and it was in Indonesia. World. Currently, as of 2020, there are 130 lakh cases per year, 21,000 to 1.4 lakh deaths per year, and the strains. So, in previous years, in 1900s, it was actually classical was first, but in 1950s, after the emergence of epidemic 7 in 1960s, the L-Tor variant has become more common than the classical, and the least common is O139 variant. In India, as you can see from 2011 to 12, 13, 14, 15, as you can see 2016 we had many cases. Originally it was confined to its state of West Bengal, which was its homeland, but as we can see it has reduced in all states consistently, and now it is mostly affected in Maharashtra, Karnataka and Kerala. Bengal was its original home state, now it affects whole of India and now the cases have reduced. And now seasonal outbreaks in India. So as you can see these green color spikes, what do you think it is? Because this is a waterborne pathogen, during the monsoon season when there is large amount of rain, there is flooding due to the inadequate infrastructure, we see spikes in cholera outbreaks. Determinants. So first, what are the reservoirs of cholera? It is humans, because it lives only in humans and we are the only carriers. So carriers for classical type. Classical type 1 is to 50 case to carrier ratio, and for L-Tor type it is 1 is to 90 case to carrier ratio. So why is it the case? Because this is due to, we will see soon. Environment, why does it come? There are many factors. There can be low sanitization, there is inadequate separation of sewage from drinking water, and then during pilgrimages like Kumbh Mela, when there is large amount of people, so there is no adequate sanitization, and in estuaries, because this is a salt loving halophilic bacteria, as we have already discussed, it thrives in estuaries which have adequate salt concentration in the level of 1%. Persistence. There have been 7 epidemics. Our body has immunity against it, but still why are we getting 7 epidemics? Because they are latent in crustaceans, seawater, planktons. So once there is an epidemic, they travel to the sea, and where they are latent in seawater and planktons. You should not get confused that it doesn't have any other, its reservoir is humans and humans are the only reservoir of it, but they can live in seawater or planktons. They can. It doesn't mean they need it. It's not their reservoir resistance. They can survive refrigeration, but they cannot survive beyond 56 degrees Celsius. They are heat and acid stable, but labile. Sorry, there is a heat and acid labile, and they are alkali stable and salt stable. These two factors, alkali stable and salt stable, are the basis for laboratory diagnosis of cholera. Okay, now we will see about lab diagnosis. So what samples will we collect? First we can collect the rectal swab, we can collect the stool specimen. So now if it is going to take more than 30 minutes, we need to prolong the viability of the specimen, because Vibrio cholera, since it is a gram negative, it is aerobic. It is very susceptible to desiccation. So we need a transport media, and also to inhibit commensals. Like the sample here, we are taking a stool specimen which contains many commensals to inhibit commensals. So what media are used? You can use a Venkatraman media, you can use a Cariblair media, and we can use the autoclave seawater. All of them contain NaCl because it thrives in NaCl. For the culture methods, in nutrient agar, as you can see, we introduce 1% salt for the inhibition of commensals. We can see mucoid colonies, many mucoid colonies. Next, TCBS agar. What is TCBS agar? It contains thiosulfate, bile salts, and sucrose. Bile salts impart this green color, as you can see. Thiosulfate, it is a nutrition for the bacteria, and sucrose, this is a differential medium. The cholera utilizes sucrose and ferments it. This fermented sucrose gains the color of yellow. Now what is the use of this color yellow? It is because we can use it to identify with other organisms like Salmonella or Vibrio parahemolyticus or Vibrio vulnificans. We can use other media like McConkey agar, which you know is for lactose fermenters. It is a late fermenter. If suppose it is fermenting very early, we can know that it is a V vulnificans is present. If alkali bile salt agar is there, it can be commensals. It is used to inhibit commensals. Blood agar, we can see that there is greenish hemolysis, but Vibrio parahemolyticus is beta hemolytic. So we can use this to differentiate between Vibrio parahemolyticus and Vibrio cholera. Enrichment broth. It is used to enrich cholera and to increase the concentration, because the stool specimen might or might not contain the adequate amount of bacteria. So we use alkaline peptone water. As we have already discussed, it is alkali stable, and peptone, it is its nutrition for the bacteria. So microscopy and staining. As it is a gram negative. As you can see, this is a gram negative, comma shaped, and as you can see, it looks like a fish in a stream. So if you have ever seen a stream, you can see that fishes keep moving like this, haphazardly and fast. So that is how they show. That's called darting motility, and it's fast and rapidly changes direction, and this is called a darting motility. So now we have found out all the culture methods, we have seen even the motility and the microscopy and the staining. Now how can we identify it? So we have automated, which is Malditoff, and Malditoff, as you know, is for laser guided, which uses ribosomes. Next we have biochemical text. ICUT test. It can be positive, this can be indeterminate, and this has to be negative, and this is acid. Because sucrose is fermented, it will be yellow in color. Now strain test. We take a colony, we use sodium deoxycholate. When we mix these two, it becomes mucoid. When we lift it up with a loop, it forms a string. Next, molecular methods. We can use PCR for PCR specific genes of cholera, and we can also use biofilm array, which is an automated method and which has 22 GI pathogens. Now moving on to the vaccination, we have three types of vaccination, and we do not give intravenous vaccination here because it doesn't cause intestinal immunity, and we give only oral as it is a mode of transmission of this disease and it is also the mode of vaccination. So there are three types. First is a whole cell vaccine. We have the whole cell vaccine combined with a recombinant B vaccine and a live attenuated vaccine. All of them are oral. So what does this whole cell vaccine contain? It contains killed 01 or O139 bacteria. This contains the same as the whole cell vaccine plus the mRNA of the cholera toxin B. As we have already seen, cholera toxin has parts A and B, so we use the B, which binds to the M1 ganglioside receptors. And for the live attenuated, it is not a killed bacteria, but it is a mutant which doesn't have the toxin. Why doesn't it have a toxin? Because the toxin is phage coded and this bacteria have been bred in such a way that they do not have this toxin. So what is the dosage? We give 2 doses for 2 weeks. This 2 doses 1 week and this is just 1 dose. For how many years does it protect you? This protects you for 3 years, this protects you for 2 years, and this protects you for 6 to 7 months. We'll get to this again. So they are used in epidemics. So all the cholera vaccines are used only in epidemics. They are not used in the inter-epidemic period because they can, as you can see, they have just 3 or 2 years of immunity. So we want it to be lifelong, so for it to be given in inter-epidemic periods. And this, when there is someone who is visiting a endemic area, he is given this live attenuated vaccine which provides him immunity for the 6 to 7 months.